afternoon, everyone. Firstly, I'm delighted to be here today, so thank you very much to the organisers for inviting me. It was great to leave a very rainy London this morning and get here into the sunshine, still this morning. So I'm going to take us back to brain tumours. Luckily, a lot of the introduction has been covered for me, which I think will help with time. So I think we've covered the initial thing. Brain tumours are obviously a problem. Astrocytoma is one type of brain tumour, and these astrocytomas are graded 1 to 4 by the World Health Organization, grade 4 being the glioblastoma. This is the most aggressive, most common, and least understood of the astrocytic brain tumours. We've been through the survival already this morning, but the prognosis is poor, and the thing that wasn't perhaps mentioned this afternoon earlier, was that actually a lot of patients don't get through the full treatment because they actually die before this. So there's actually desperate need for improved treatments in this brain tumour group. One really good potential target or therapeutic target is a cancer stem cell in these brain tumours. And I'm going to offer a definition of a cancer stem cell now. A cell within a tumour that possesses the capacity to self-renew and to cause the heterogeneous lineage of cancer cells that comprise the tumour. So what is the evidence for cancer stem cells generally? As I've mentioned, it's a very attractive hypothesis. In 1997, there was some good evidence of cancer stem cells being present in leukemia, particularly acute myeloid leukemia, with only CD34 positive and CD38 negative cells being able to transfer acute myeloid leukemia into immunodeficient mice. With regards to solid tumours, the evidence for cancer stem cells has been a bit more variable. In breast cancer, there is some evidence of cancer stem cells, with CD44 positive, CD24 negative cells being found to be tumour-forming in vivo. However, other tumours, such as melanoma, there's not really any evidence for cancer stem cells, with all cells being tumour-forming. So what about cancer stem cells in glioblastoma? The marker that's been most looked at is CD133, and another marker that's raised its head is CD15, which obviously is the basis of this talk. So I'm going to start with CD133. It's a surface antigen, which is found to be a marker of embryonic neural stem cells. It's expressed on human, fetal, and adult hematopoietic stem cells and progenitor cells. And it was initially thought to be a good cancer stem cell marker in glioblastoma. Initial research showed that it was tumour-forming with a very small numbers of cells when implanted into mouse models. And also, it was able to differentiate into the full neural lineages. However, more recently, evidence for CD133 has really been put in doubt. It's been found not to be present in every tumour sample of glioblastoma. And it's been found that both the positive and the CD133 negative cells are tumour-forming. So this is why we were interested in CD15, which the immunologists amongst you will know is also expressed on leukocytes. This is a carbohydrate moiety. It's diversely expressed, but focused on stem cells, so this makes it an attractive target. It's been found to be expressed on the surface of B stem cells and C transient amplifying cells in the mouse subventricular zone and hippocampus. So again, it's showing that this marker is expressed in neural-type stem cells. So how about in brain tumours? There was some initial research looking at this marker in 2009, which showed that CD15 positive cells were more tumorigenic than their negative counterparts and had a high level of expression of markers associated with neural stem cells. So this made it an attractive marker to investigate further. So our aim of this study was to determine if CD15 did represent a credible stem cell marker in glioblastoma, and to find out whether this potentially could be a therapeutic target. I'm purposefully not giving away the punchline yet. So, in terms of methods, we used patient samples and we used in vitro cell lines. I'm going to run through how we generated our in vitro cell lines in more detail because I think the methods are quite important. And we also used in vivo animal models. And for this research, we used the nude mouse model. So on to the glioblastoma cell lines. There's been some sort of controversy in the glioblastoma field as to whether it's better to culture stem cells in a serum-containing medium or a serum-free medium. The cell line that was mentioned earlier, U87, is a cell line that is actually cultured in a serum-containing medium. But it's been found more recently, particularly in a paper by Lee et al. In 2006, that the genotype more closely resembles the parent tumour in serum-free media. So serum-free media, our serum-free media, does contain some growth factors to assist with the proliferation of our cells. So this is a method that was derived in the lab in which I did my PhD. First of all, we take the fresh human patient samples directly from the laboratory. We mechanically dissociate and enzymatically dissociate the sample and allow the cells to grow as a neurosphere. And the really reason for this first stage is to remove the debris and select proliferating cells. When the neurospheres, which is exampled in A, have grown to a certain size and they look healthy but aren't actually becoming necrotic in the centre, we allow these to grow as a primary monolayer using extracellular matrix to coat the flasks. And then subsequent passages conducted as a monolayer. So this culture method is quite useful for us. It's a combination of suspension and adherent culture. Obviously, a monolayer doesn't have the cell-to-cell interactions that you would have in the 3D system of a neurosphere. But it has distinct advantages that you can follow individual cells in the adherent culture. It's possible to screen many different tumours using this method. All of our cell lines thus far have been confirmed to be tumorigenic in vivo in mouse models. And so far, our success rate has been around 92% from each tumour sample to generating a cell line. And this is even accounting for the fact that some of the samples are from the very necrotic core, which is so much a feature of glioblastoma. We have also demonstrated that primary cell lines were cytogenetically similar to both the parent tumour and the experimental xenograft. So this is an example, and this is using SNP array, which has been nicely described earlier today as well. And as you can see here, the parent tumour, the cell line, and the xenograft are actually remarkably similar at early passage. So these are taken from passage 6 in one of the cell lines, and this has been repeated in triplicate. So what about the results? I'm going to start by talking about the patient sample results. So the first thing we did was we looked at patient samples, sliced tumour samples. You can see a representative hematoxylin and eosin staining of a patient sample at the top. And then we looked to see how readily CD15 was expressed. As I've already mentioned, CD15 is also expressed on leukocytes. So we obviously didn't want to include this in our research. So we only included CD15 positive and CD15 negative cells that also co-expressed GFAP, a marker of astrocytes. And what we found was that CD15 positive cells were actually very rare in our samples. We looked at 10 different samples. On average, only 0.11% of cells were CD15 positive. And then we looked to see whether the CD15 positive cells were proliferating or not. Obviously, if this was a stem cell, you may feel that these cells would be likely to be the proliferating ones in the tumour. And we actually found that they weren't regularly proliferating. I think we only found about 3 cells in all of our 10 samples that were CD15 positive, GFAP positive, and also labelled for Ki67. However, in the non-CD15, so the CD15 negative cells, the cells were regularly proliferating, about 5.5%. So next, having found that actually these cells weren't really, the CD15 positive cells were rare and rarely proliferating, we wanted to see what happened in vitro in our cell lines. And you could arguably say we're selecting for a more proliferating population by our methodology. The first thing we wanted to do was to look at the cytogenetic profiles of sorted cells for CD15 positive and negative. So we fluorescent activated cells sorted CD15 positive and negative and looked at the cytogenetic profiles using SNP arrays. And as you can see here, both the positive and the negative cells were the same using a SNP array. So for example, if you look at chromosome 7, chromosome 13, there's a large, firstly there's trisomy, both seen in the positive and the negatives, and there's an area of loss of heterozygosity as well. Next, we're interested to look at gene expression profiles. It's all very well looking at the chromosomes, but why don't we look at the genes that are expressed? And we did a gene expression array looking at sorted CD15 positive and negative cells and found that there was no significant difference in gene expression between these two cell populations. So this is getting a bit sort of sad for CD15, unfortunately. So we next looked at in vitro cell lines and looked at staining for markers of neural stem cells and neural progenitor cells, really trying to repeat the work that's previously been done. And we found that there wasn't a significant difference in expression of either neural stem cells or neural progenitor cell markers between CD15 positive and negative cells. So we looked at nestin and SOX2, which are neural stem cell markers, and OLIG2, PDGF receptor alpha, and NG2 being markers of neural progenitors. And there was no significant difference in the expression of CD15 of these markers in CD15 positive and negative cells. So what happens if you sort the cells? Do you manage to retain the positive and the negative populations separately over time? So we, with several different cell lines, sorted the cells for CD15 positive and negative, again using FACS sorting, and looked over time at the CD15 expression. And what we found was that actually quite quickly the CD15 negative cells here were actually able to generate cells that were CD15 positive, and also that the CD15 positive cells rapidly generated cells that were CD15 negative. And this was seen in all three of our cell lines in which this was assessed. So we next thought, well, maybe there's a chance that if you look at single cells, perhaps if you sort cells singly for CD15 positive and negative, there might be some differences. So we seeded single cells into 96-well plates and allowed them to proliferate for 6 weeks, and then looked at the number of CD15 positive and negative cells, again by flow cytometry. We found that there wasn't a significant, that the positive cells were able to recapitulate a population with positive and negative cells, and that the negative cells were also able to develop some positive cells over time. So this showed that there wasn't any hierarchy between the CD15 and made it obviously less likely to be a cancer stem cell marker. So we're interested to see whether perhaps the CD15 positive cells were more able to form colonies in vitro, and we found that there was no significant difference again. Both the positive and the negative cells were equally colony forming in vitro. So as a final ditch attempt to try and get sort of a difference between CD15 positive and negative cells, we actually tracked single cells over time. So we looked at single cells implanted into 96-well plates and watched the colonies grow on day 2, 4, 7, and 12, and literally counted the cell numbers in each colony, and again found that there was no significant difference in proliferation of these single cells over time. So far, we've found that CD15 positive cells are rarely expressed in the patient sample, and in the patient sample, they're rarely proliferating. And we have also found that there's not really any differences between the positive and the negative cells in vitro. So what happens when you implant them into animal models? So we used nude mouse models, as I said, for this, and we implanted positive cells into the left flank of the mouse subcutaneously, and negative cells in the right flank of the mouse, the same mouse, to try and keep everything as similar as possible. And we again found that there was no difference. So both CD15 positive and negative cells were equally tumour forming. And we looked at the sizes of these tumours and couldn't find a difference in the tumour size either. So in conclusion, I think, sadly, CD15 does not identify a subpopulation of cancer stem cells and isn't able to assist us with finding a new therapy for this brain tumour that really needs one. However, I think an important thing is that we have a model here that could potentially be used to find either patient-specific glioblastoma targets or alternatively just to look for further potential molecular markers. Obviously, this work was done by a lot of people. It was under the supervision of Colin Watts and Phil Jones. Colin Watts at the Brain Repair Centre and Phil Jones at the Hutchinson MRC in Cambridge, and also with the help of Joe Fowler, Talal Alfile, Sarah Piccarello, and Inma as well. So thank you very much.
It was great science. It was a negative result, but a very well documented.
Yeah, I'm sorry. I know it's a bit depressing presenting negative results, but
you know, if it's well documented and it addresses an interesting question, an important question, negative results should be as violent as positive ones.
But they're not.
They're not. That's the problem. But they should be. Really. Are there any questions?
Behind that, if you would address therapeutically the stem cell, you would have an easier target to eradicate the tumor?
Yeah, so
It seems not likely in view of the heterogeneity.
no, and I agree with you, and I think that gradually in brain tumours, and certainly as a lab group, we've moved away from trying to investigate cancer stem cells and are now dealing with the glioblastoma as a heterogeneous population, as you said. The attractive thing is obviously, if you discount brain tumours, but in other cancers and brain tumours to an extent, you get recurrence. And I think the idea is that if you were able to target a cell that was capable of recurrence and reforming the whole tumour, that would be a very important development. However, if you can't find a marker for a cell that's able to recapitulate the entire tumour, then actually you're sort of stuck. But as you say, I think more likely, I think the general movement in the field is moving towards actually that we do have a heterogeneous population particularly within glioblastoma. And there's even some evidence now that different areas of the tumour are very different in terms of gene expression. So I think that probably there may not be such a thing as a cancer stem cell.
Are there any types of solid tumors or leukemias where cancers, where stem cells have been documented with rigorous methodology, like or is it still a vague hypothesis?
I think with everything, and the difficulty with this work was that actually someone had documented that they had found CD15 to be a potential cancer stem cell. It's very difficult to document, you know, really rigorously. I think partly because it's very difficult to always get the markers and the antibodies to work very well. There has been some evidence in breast cancer that's looked fairly good for that, looking at CD44 and CD24 negative cells. But I think it's difficult to have a really robust marker. And also, people often find that because there's heterogeneity within the disease even, it's difficult to find a marker that's robust throughout every type of even one cancer.
Could you argue whether it's not totally impossible? Because usually the clinical situation, when you have the tumor samples you actually have access to, is a sample that is relatively old. I used to work on ovarian cancer, and it's all very old. The cancer is very advanced. To find such a cell is probably possible when you have a tiny little tumor, the originator perhaps the size of a needle pin or something like that,
Yeah, no, I agree with you, but I think it would be important if there are what seems to be basically, unless you can demonstrate that every single cell of a tumour is capable of forming a cancer. So, for example, if you implant it into a mouse, and this is something that's been shown in glioblastoma, you can actually take a single cell, implant it into an animal model, a very immunocompromised mouse, and you can develop a tumour in nearly all of the cases, which is quite unusual. But it's very difficult. I think it does seem that some cells within the stochastic environment are more tumour-forming than others. So I think perhaps if we can target these cells, potentially we'll have reduced recurrence. But I mean, it's very difficult to say.
I was just wondering if the observations you've seen and you've reported have anything to do with the, yes, tumors are heterogeneous in nature, but part of the problem with respect to their growth in vivo is that they grow in 3 dimensions and they subsequently become more hypoxic, which is one of the contributing factors why they're so difficult to treat, certainly in response to radiation therapy. Is that a plausible explanation for some of these phenotypic expressions based on the fact that you have several different biochemical processes that are taking place inside a tumor in vivo, which may or may not be expressed in vitro unless they do express a change in their hypoxic cell state in vitro as well?
Yeah, I agree with you. I mean, it's difficult to find an ideal model in vitro, obviously, but I think the fact that in animal models as well, where you do have though you don't have the same, it's not obviously the same as human, but you do have a 3D environment, the fact that you've got everything's, they're the same, and that is quite convincing. I did all this in normoxic conditions. Obviously, the centre of a brain tumour isn't normoxic, but I think it's very difficult practically to actually take a tumour from surgery where obviously it's going, you have to perform surgery as you normally would at the moment to get ethical approval. And so it's difficult because they've already had a hit of oxygen as they're extracted from the brain.
Right. I was wondering, is it possible then to culture the cells in an anaerobic environment like you would in certain types of bacteria?
Of course you can, but you can't get away from the fact that you're going to take it out of the... So it's already got... Do you see what I mean? You can. Obviously, you could do that. It's not something I've looked at, but I think other people have looked at it to some degree or other. And actually, interestingly, when you were looking at the mRNA, the expression in U87 in the last glioblastoma talk, actually, the hypoxic conditions weren't perhaps that hypoxic then, if you see what I mean, compared to normal. I think it's very difficult. I think there's lots of things you can do. I think this is a fairly robust model, partly because of some other research which I'll happily talk about later. But there's no perfect model, is there, in vitro at the moment? And if we could find one for all cancers, then hopefully we'd be able to find therapies, and that would be...
Exactly. Thank you.